Well, friends, let us now study high pressure processing of foods. This is organized in two parts. In part 1, we shall be discussing about the process principle and technological aspects of high pressure processing, whereas the part 2 will include effect of high pressure process parameters on food constituents and its quality, as well as we will also discuss the application of high pressure processing in various foods. High pressure processing is a novel method of food processing. It is basically a non-thermal method. It is receiving worldwide attention now and is a new concept compared to the conventional thermal processes. High pressure processing is also termed as hyperbaric pressure, ultra high pressure, high hydrostatic pressure processing or Pascalization. What is done in high pressure processing? Actually, in this process, the food is subjected to elevated pressure of up to 900 megapascal, or in other words 9000 atmospheric pressure, with or without the addition of heat, to achieve microbial inactivation, enzymatic inactivation, or to alter the food attributes in order to achieve desired qualities in food. As far as the principles of the process is concerned, there are basically 3 principles are involved in high pressure processing, and those are the isostatic principles, Le Chatelier's principles, and principle of microscopic reordering. So, isostatic principle states that application of pressure is instantaneous and uniform throughout the sample, as you can see in this figure. From all the sides, everywhere inside the sample, the pressure will be uniform. Le Chatelier principle says that there is a reaction volume change during high pressure processing, and this change in reaction volume is influenced by high pressure applications. Those reactions which result into the volume decrease, they are accelerated with the application of high pressure and vice versa. The principle of micro reordering says that at a constant temperature, increase in pressure increases the degree of ordering of molecules. Therefore, pressure and temperature exert antagonistic forces on molecular structure and chemical reactions, which depending upon the pressure and temperature of the system, it influences the various characteristics of the food material. As far as the technology of high pressure processing is concerned, normally the food material taken in a packaged form, obviously in the sterilized packaging materials, this packaged food material is loaded in the pressure chamber of the high pressure equipment, and then the desired pressure is applied, the food material is held at the desired pressure for desired duration of time, and then the pressure is released, that is it is depressurized. So, the food in packaged form first pressurized, held at the desired pressure for desired time and then depressurized. Okay. This is actually the technology of high pressure processing. You can see here in this equipment, that is schematic diagram is here of high pressure, that is it has a pressure chamber, it has the arrangements to generate the pressure by a suitable type of pressure pump, and then that is it has been the instrument is provided with the arrangements for heating, cooling and complete or proper sealing of the pressure vessel. Regarding pressure generation system in the high pressure equipment, there are two types of systems. One is direct compression system, another is indirect compression system. In the case of direct compression system, as you can see in this figure, that there is a fluid or the compression fluid, which is normally water, is loaded into the pressure chamber, and then pressure vessel is closed at the bottom, and from the top normally. Okay. It may depend upon the even system, from some cases it may be the pressure, but held from the bottom also, but in general in majority of the equipment which are available, there is a piston arrangement with the help of a low pressure pump, that is the water inside the chamber is pressurized. So, that is the direct compression system, that is the fluid is compressed and this compression generates the pressure. In case of indirect compression systems, the fluid is pressurized somewhere else, that is pressure medium tank, and then this pressurized fluid is loaded in the pressure chamber of the equipment through some or with the help of some intensifier. These are high pressure pumps, intensifier, and then this seals etc. are closed. So, this is basically two systems which are used in the commercial equipment of the high pressure processing. In this picture, we have shown that a photograph of a high pressure system, that is the, and just to make you understand what are the different components in a high pressure system. Obviously, as you could see in the earlier diagram, that is it has a pressure vessel, the equipment should have a pressure vessel of desired capacity. Obviously, there should be proper closures and sealing, there is arrangement to close and seal the vessel, and proper device for holding the closures in place while the vessel is under pressure. For example, yoke etc. could see here the yoke, that is it should keep the closures intact, so that the inside the chamber is completely sealed. And then depending upon the system, we should have higher pressure intensifier pumps, pressure and temperature controlling and monitoring system, and also particularly in the continuous system, product and material handling system. And of course, the material of construction of these equipments should be able to withstand the high pressure generated inside the chamber. Since in the high pressure processing, the food is generally packaged in its final form and then treated, and these packets are opened only at the consumer end in order to avoid any recontamination etc. Normally, they are opened just before the consumption. So, obviously, that this should be a system, that is the packaging material, its requirement should be that which can withstand, number one, that the packaging should be able to withstand, material should be able to withstand inside the pressure chamber, that is such high pressure generated. And also you have seen in that Le Chatelier's principles that there is a some sort of decrease in the volume of the component of the sample as a function of pressure during the compression, and almost an equal expansion occurs when the material is recompressed. So, obviously, the packaging material should be able to withstand these changes. So, in fact, it should be flexible packages, airtight flexible packages that can withstand a change in the volume corresponding to the compressibility of the product are needed here in this system. Also, in general, it is seen on an average there is 15% reduction in volume and similar of course, return to its original volume without the loss of barrier properties, means that is the packaging system must be able to accommodate about 15% reduction in volume and it should return to its original volume after decompression without any loss of the barrier property. That is important. That is, in this process of changing the volume, reducing the volume, increasing the volume, the barrier properties of packaging material should not get destroyed. So, airtight packaging material and of course, it should be impermeable to oxygen. It should be opaque to light, particularly when we desire to retain the fresh color and fresh like flavor of the high pressure treated foods. Because the oxidation reactions normally in the high pressure processed foods, they generally tend to change the material characteristics, that is, so the oxygen and then light which is normally it catalyzes the oxidative reactions. So, this should be arrangement should be such that these reactions are minimized. So, accordingly the packaging material should be impermeable to oxygen, it should be impermeable to light etc., opaque. So, the plastic films normally EVOH, that is ethylene vinyl alcohol or polyvinyl alcohol PVOH copolymer films are normally used, they are better suited to high pressure processing. Even sometimes depending upon the material characteristics, semi rigid containers can also be used. Vacuum packaged products are ideally suited for high pressure treatment, but mind it, metal cans and glass containers are generally not used, obviously because of their rigidity. Glass containers or metals, they can break during the compression state. So, this the use of these material should be avoided, it is not recommended. In this slide, we have just seen that as far as the process or product application is concerned. If you look in the international market, high pressure processed products are available commercially. Those products generally are jams, jellies, juices, salad dressings, fish, meat products, sliced ham, rice cake, yogurt, etc. Of the total high pressure products available in the global market, on an average basis 32% proportion is of vegetable products, 27% on an average basis is meat products, about 16% constitute the high pressure processed products of seafoods, juices and beverages allow 11% and other products are 14%. The various people have worked different process combinations, that is combination of pressure and temperature and time are reported in the literature depending upon the requirement of the process. As you could see here in this figure, in general, that is for improving the material functionality etc., for the extraction processes or other improvements, generally pressure in the range of 100 to 300 megapascals. For comparatively lower holding times are used. About pressure in the range of 400 to 600 megapascal, little higher holding times are recommended, are used commercially for pasteurization purposes. If intention is to sterilize the product depending upon its acidity, other factors of the food, even high pressures like 700 to 900 or even sometimes more megapascal are required for even higher holding times. So, this gives an idea that is one has to select the pressure requirement depending upon the characteristics of the food materials, its various components, and what are the desired application, for what purpose this high pressure processing is being used. Let us see what are the pressure temperature effects in a high pressure system. When we apply the pressure in the food, that is the another influence, that is temperature. What are the different interrelations in the high pressure as well as temperature in the food. So, I hope you all know that temperature increase of the food material under pressure depends on factors like the final pressure used, that is at a lower pressure the temperature increase might be less, at higher pressure obviously the temperature rise will be more. Also the product composition and even the initial temperature of the material or initial temperature of the compressing fluid, that influences the overall rise in the temperature when the food is pressurized, or further reduction in the temperature when the food is depressurized. In general, it is seen that the water, there is a increase of temperature by around 3 degrees Celsius for every 100 megapascal increase in the pressure at room temperature. Both pure water and most of moist foods, and they are subjected to a 600 megapascal temperature at ambient temperature, will experience about 15% reduction in volume. So, these are the important considerations in the processing. You can see here in this figure, that is the compression phase, holding time and decompression phase. So, during these periods, how with the changing the pressure, how the temperature of the material changes. You see here, that is T 1 to T 2, it is known as the compression phase, that is from the initial pressure which is P 1, the pressure is raised to a value P 2, right, and as a result of this increase in the pressure, all right, depending upon the rate of the compression, that is whether it is pressure is applied very fast, pressure is applied very slow, there is certain increase in the temperature, that is from initial temperature T 1 to it reaches to T 2, and of course, this T 1 from T 1 to T 2 obviously it may depend upon the compression rate and the compression time. Then this time T 2 to T 3 is hold time, or also commonly known as dwell time. So, means during this the food material is held at that temperature, that is P 2 and P 3, there is a straight line, means there is at a constant pressure, and during this period temperature also it remains almost constant. There might be very slow decrease depending upon the system, depending upon the perfection of the pressure, depending upon the other constituent, and then finally T 3 to T 4, it is decompression phase, when the pressure is released, all right, it comes to its original value of P 4, and then the temperature also from T 3 it lowers down to T4, all right, and this temperature T4, it is slightly lower than its initial temperature T1 because of the heat loss during the compression period. So, because of this heat loss, final temperature of the decompression is generally little less than that which was there in the initial stage. Let us see how when you compress, when you put these foods under high pressure, how the microbials or microorganisms get inactivated, get killed, because that is the main thing of that when we want to extend the shelf life using high pressure. So, microorganisms or enzymes etc., they must be inactivated. So, the inactivation of microorganisms during high pressure involves shear force generated membrane disruption. This results into interruption of cellular function, localized thermal damage and protein deformation. Means that the routine physiological processes of the microorganisms are disturbed and obviously the microorganism gets killed or inactivated. You can see here in this figure, before the process it was the shape of the microorganism, when the pressure is applied from all the sides, there is microbial cell compresses. Depending upon the pressure, the compression will be more or less, and then after it is decompressed, it may again come to its earlier size, but here its activity is guessed, because you can see here the material it has decompressed, it is oozes out. So, it is no longer physiologically active. As far as the resistance of microorganisms to the high pressure treatment is concerned, generally spores are more resistant to pressure than their vegetative cells. Even spores of gram positive cells are more resistant to that of the gram negative cells, and in general that is spores have higher resistance than gram positive cells and then gram negative cells. And of course, pH and water activity of the food affect the lethal effect of the pressure during high pressure processing. Let us little elaborate, that is effect of the high pressure on bacteria. The high pressure influences both, that is pathogenic as well as spoilage microorganisms, that is both can be destroyed during high pressure processing. However, there are large variation, see that is the particularly the pressure resistance, baroresistance of the microorganism, all right. It varies in different bacterial strains. Nature of the medium in which the pressure is applied, that also affects the response of microorganisms to pressures. The cell wall of gram negative bacteria are significantly weaker than that of the gram positive bacteria, and therefore, gram negative bacteria are more pressure sensitive than gram positive bacteria. Bacterial spores, obviously as I told you earlier, they are more resistant to the bacterial cell. High pressure up to maybe 800 megapascal are needed to kill bacterial spores at ambient temperature. Even spores of certain bacterial species might need as high as 1400 or 1500 megapascal for killing or inactivation, particularly in low acid food at ambient temperature. This pressure induced inactivation of bacterial spores is significantly enhanced with the temperature, particularly if the temperature is 50 to 60 70 degrees Celsius, and perhaps it is also at or below 0 degree centigrade temperature. So, the best thing is that the combination of pressure and temperature, that is the pressure at lower side maybe 50 to 300 megapascal is needed for the germination of the spore. So, when you apply this lower pressure comparatively 50 to 300 megapascal, the spores can germinate, and then once they are germinated, they can be easily or relatively inactivated by relatively mild heat treatments or even higher pressure treatments. So, it is always advisable to have a combination of that is the pressure as well as temperature to inactivate spores, particularly in low acid food. Of course, while doing so, that one should see the effect of temperature and the combination of temperature and pressure on the food characteristics. Regarding the effect of high pressure process parameters on the fungi, these fungi they are normally lesser resistant. About a pressure of less than 400 megapascal for a few minutes is sufficient to inactivate most of the yeasts. At about 100 megapascal, the nuclear membrane of yeast is affected. At pressure 400 to 600 megapascals, alterations occur in the mitochondria and cytoplasm of the yeast cell, and of course, it ultimately causes the destruction of its normal activity and inactivation. Pressures between 300 and 600 megapascal can inactivate most of the molds. Sensitivity of the microorganisms, of course, during high pressure processing is the effect of that is the microorganism, that is the various factors influence the response of microorganism, including pathogens, to pressure treatments, and obviously these factors must be considered while in optimizing a high pressure process parameters for the microbiological safety. The major factors like that in the case of thermal processing, almost similar factors here also influence, that is the pH, water activity, temperature, pressure or holding or dwell time are the important factor which influence the sensitivity of the microorganisms to high pressure processing during high pressure processing. And you can see here in this table, that is some people have reported that our high pressure treatment required for 5 log cycle inactivation of E. coli and Staphylococcus aureus in poultry meat and milk during a 15 minutes treatment. That is, with the increasing temperature, you can say up this the pressure requirement for 5 log cycle reduction decreases in both in the case of poultry meat as well as in the case of milk. Of course, the pressure requirement for the similar inactivation in milk is more than that of the poultry meat. This is for the Escherichia coli, and the same trend is also for the Staphylococcus aureus. Other table you can see, that is another gentleman, their team, they have worked for similar 5 log cycle reduction in the inactivation ratio of certain microorganisms. They have studied during a 15 minutes process, and they found that among the different groups of problematic microorganisms in the food, Staphylococcus aureus under their experimental conditions were found to more pressure resistant than that of the other microorganisms study. Another, that is the like microorganisms, enzymes are also sometimes they result into the deteriorative reactions, and their inactivation becomes another important criteria for the high pressure stabilization of the food. In addition to microorganisms, your process should also be able to destroy certain enzymes. Okay. And in these figures you can see how that is the del G is a function of pressure. Changes in the delta G will be there, that is the whether this protein remains in its native state or it's is a denatured, is a function of both pressure and temperature. Normally, it has been seen that the primary structure of protein generally not destroyed or not affected by high pressure treatments. Tertiary structures are modified, followed by a large hydrogen change. disruption of hydrophobic and electrostatic interactions during high pressure processing may lead to certain changes in the quaternary structure, and there is a dense hydrophobic hydration layer is formed, which will lead to the volume changes. So, these are the effect, means that is by changing in the conformation of the structure etcetera, denaturation, that is the another changes, the activity of the microorganism is influenced, and under the extreme conditions when the pressure is relatively high, the enzymes may get inactivated completely or their activity might be reduced. Here in this table you can see various people have worked and they have seen the stability of the various food enzymes like pectin methylesterases, peroxidase, alpha amylase, lipoxygenase and polyphenol oxidase. And it is seen that as depending upon the again pH of the system, water activity of the system, temperature present and other system, there is these studies which were conducted on various high pressure ranges and temperature ranges, and it is found in general that the polyphenol oxidases and peroxidases are comparatively stable to pressure treatment. Sometimes they may require higher side like 800 to 900 megapascal to pressures to for their inactivation. Other enzymes like pectin methylesterase enzymes are also equally stable. Of course, they need lesser pressure, and the pectin methylesterase enzyme, and it was comparison was made in their activity in the tomato as well as in the orange, which differ in their acidity, even their activity also differed. So, friends, you have seen now that is what are the basic principles on which the high pressure system or high pressure process is affected. The major deteriorative agents like microorganisms, enzymes etcetera, how they are inactivated and what are the different systems which are used, machineries etcetera, which are used for high pressure processing of the food. Thank you very much.